we were talking about emotion, and we ended Tuesday with talking about all of the theories of how emotions are generated. What emotions are? Are they physiological? Are they interpreted by our conscious awareness? What do they have to do with our facial expressions? So on and so forth. Okay. And I ended with the last two being like the evolutionary theory, which is basically an amalgam of all of the things that preceded it, and then facial expressions being mostly an evolutionary idea, right? Because if it's a human universal, then it's then it's an evolutionary thing, right? Regardless of culture and environment and things like that, we all evolved to we being people have all evolved to have these facial expressions even when we don't want to have them, because we talked about leakage, right? So today, what I want to focus on is the brain stuff, and so we're going to start with the we're going to start with the amygdala, because that's by far the most important brain structure for the many of the emotions that we do feel. We are going to focus a little bit more on fear than any other emotion, just because like I said, most on Tuesday, most of the research has been done with fear. Okay. There are three complexes. We're essentially going to speak about two consistently over the course of the next few slides. But there are three complexes within the amygdala. The amygdala sits at the very end of the basal ganglia, so at the very anterior portion of the basal ganglia, and it's firmly connected to the hippocampus. So it's right next to the hippocampus. They share a lot of connections. You can see that connection right here, right? So it kind of looks like this. A lot of illustrations just make it a ball shape. So a spheroid. This is This is more teardrop shape, I suppose. But you can see all of the connections it has. So these are all brain areas, okay? These are all like cortical areas going above it, essentially. And then these are all areas that are below it, quote unquote. And what I mean by above and what I mean by below indicates the streams. So dorsal and ventral streams, right? So dorsal is cortex, and ventral is like midbrain, hindbrain stuff. So things that you don't have conscious awareness of. So, you have your sympathetic activation, parasympathetic activation, reflex potentiation, all of that, stress hormones. These are all things that you do not have conscious awareness of. Everything up here, it has to do with things that you could potentially be consciously aware of, right? Our memories into the entorhinal cortex and the perirhinal cortex, right? These are all structures associated with the hippocampus and memory, right? And then we have our auditory association cortex. So those are things we hear. And we will talk, the connections with the auditory cortex is actually really strong with the amygdala for things like startle reflex and other auditory things that bring out emotion. And you can see that the emotional stimulus here, goes into the thalamus and then all the way through. And you can see then the arrows, auditory cortex, goes into the lateral, which is sometimes referred to as the basolateral. So this particular breakdown I got from the internet. It's not from your book. So it's using slightly different slightly different language, but the basolateral nuclear complex strengthens encoding, right? So that's where a lot of information comes from first, and that's this lateral nucleus right here. And then we have the basolateral nucleus, so these two are connected, okay? So we have then the central medial complex, which is this right here. I think I just referred to it as central elsewhere in the slides. I'm not entirely I don't entirely remember. I only made these on Monday, but shoot. It's been a long time. Central here gets information through the other side, the basolateral over here, from the hippocampus and the subiculum. Okay, and then the central then sends information to all of these autonomic functions here, right? So the central is top down. Okay, top down. Send the information to autonomic functions because I've received information from the cortex, okay? But these are autonomic functions, and so you don't have conscious awareness of them, okay? Now, there is a small complex which gets input from the olfactory bulb directly. The olfactory system is the only one that doesn't go through the thalamus. It goes straight to the amygdala, and it goes into this very small, it's not represented on this image here, but it goes into a very small complex within the amygdala, and that's where you connect smells with memory. And that you probably are aware of that, okay? Probably aware of that because you smell something, and you're like, oh my gosh, I remember this. Okay. Now, one thing that you'll notice about the size of the amygdala, I mean, there's nothing really indicating size in here, but the size of the amygdala from animal to animal to animal is correlated with how much that animal focuses on the eyes of other animals, okay? So, we have a very large, we being humans, have a very large amygdala size ratio compared to our brain because we focus on the eyes of other humans as well as other animals. We look into their eyes. And then you can move down the quote-unquote food chain and see the size of the amygdala compared to how much that animal then focuses on eyes. And so you'll start to notice the difference between prey and predators with respect to amygdala size. It's actually a really cool, interesting finding that I wanted to share with you all. So there is one more theory of emotion, but this one has to specifically do with the amygdala. And so LeDoux determined through a number of different studies, LeDoux being a cognitive neuroscientist and somewhat a behaviorist, determined that, or at least theorized, that the amygdala has two routes for processing, okay? And so your book has the continuing example of the researcher seeing the bear, right? And he said that there's a high road, which is fast, and a low road, which is slow, okay? And the high and the low road has to do with, the high road and the low road have to do with essentially where they are in the brain. So high road being dorsal, low road being ventral. And so then we take the slow and the fast. So this is the low road over here, and this is the fast road, okay? So I'm going to do the high road very quickly, okay? These are autonomic functions, okay? Fast hardwired defenses activates fight or flight response, which engages a defensive behavior, running, okay? The slow road, on the other hand, has to do with the cortical responses, okay? The cortical responses. Slow cognition. Now, this isn't, like, actually slow, but it is relatively slow when you compare it to this side over here. So this is slow, it's fast, but it's relatively slower compared to this side, okay? Because our body engages activations and processes that we cannot think about. We cannot think about them. We do not think about them, and we cannot think about them, okay? Because they are autonomic, right? And so, this is such a silly example, but slow cognition. Whoa, that looks suspiciously like Ursus Arctus horribilis, which I've learned are dangerous. Seriously, I'm sure you're not thinking about the specific species name. The species and subspecies, by the way, because there's two. Anyways, it's a bear. It's ursine. Okay? Yeah, you see the bear and you're like, ah! I'm scared! But you're thinking about being scared as you're running away. So that's what LeDoux said, but he specifically said that this is all routed through the amygdala, okay? And he was he was one of the first to take older theories and apply it to what was known about the amygdala at the time. So this was like the 1970s, okay? And so he was basically mishmashing neuroscience with behaviorism and like fear conditioning, that sort of thing, and cognition. And he was just like mashing them together. And so this is what we got. Okay, this is what we got. Further evidence comes from an issue, a syndrome called Klüver-Bucy Syndrome. You may have heard of this. And so this, and I'll let this video speak for itself. But Klüver-Bucy Syndrome is an interesting set of issues. And it's a syndrome because it's just a collection of symptoms. And you'll see why the amygdala is so important for the symptoms that are expressed by having damage to the amygdala. And this video specifically talks about humans. The interesting thing is that because monkeys have smaller amygdalas, if you damage their anterior temporal lobe, so the front part of the temporal lobe, where the amygdalae are, they have similar issues. So, excuse me. So, I'll explain what happens to monkeys, and then I'll let the video talk about humans. So this is what happens when you lesion, through lesion studies, monkeys' anterior temporal lobes, not medial temporal lobes, anterior temporal lobes, where the amygdala is housed, okay? So they increase their sexual activity, okay, beyond procreating. Sometimes this increase in sexual activity is violent, okay? And a lot of the times it's directed toward inappropriate objects. So inanimate objects where they will just try to have, where they will try to copulate with like a stuffed animal or a block of wood or something like that. They'll show a considerable and marked lack of fear if you compare it to non-lesioned monkeys of the same species, okay? They lack discernment with regard to food items, so inability to determine whether or not something is food. They'll try to eat it instead of at first realizing that it's food and then trying to eat it, like my dumb dog. And then an inclination to investigate objects with their mouths, which is interesting because they're monkeys and other primates are very handsy. We have because we are primates, we have a lot of receptors in our fingers. We don't necessarily need to use our mouths, especially when we advance in age. Yeah, babies use their mouths because we have a powerful touching instrument in our tongue, right? It has a lot of touch receptors. But then we get better with fine motor movements of our hands and our fingers, and so then we explore things with our hands. Well, when you lesion out the amygdala in these monkeys, they start to explore things with their mouths again. So it's essentially they regress away from what the hands are doing and what the hands can do. Okay. Julia, there was a case in Australia where beetle populations went down because the males were trying to mate with beer bottles. Ooh, that's interesting. I wonder, I have no idea what their brain structures are like. Very, very, very thin brain structures. But that is really interesting. Email me a link to that story if you can find it. I want to read that. Okay. So let's listen to these folks talk about Klüver-Bucy syndrome in humans. And you can see hyperorality, right? Or already there in the thumbnail. Hyperorality. So that's touching stuff to your mouth, as I just did. Aww. So it really isn't. So there's a lot of these kinds of videos on Klüver-Bucy syndrome, but I wanted to, as many of you know, I like to show stuff from popular media regarding some of the psychological stuff we talk about. So, I mean, you're welcome to go, look at these kinds of videos if you want to learn more about Klüver-Bucy syndrome. There are, as far as I'm aware, I could not find any good representations of somebody who was filmed with Klüver-Bucy syndrome. So, I mean, it might be out there, but with I'd probably searched for about 10 to 15 minutes looking for it, watching videos here and there. There's one with a quick discussion with a woman who apparently is famous for having Klüver-Bucy, but the video is mostly her dad talking about being part of a research study, and I'm like, no, I don't care about you. I care about her. I want to listen to her. So among the issues that humans have when the amygdala is lesioned, and as you heard from the character in the house clip, he specifically said lesions to the temporal lobes. I know he doesn't say anterior temporal lobes, but that's essentially where he was pointing. So visual agnosia, we've discussed that already. Failure to identify objects, loss of normal fear and anger responses. So a reduction in fear response, so less fear. As well as a reduction in anger responses, which is quite interesting. A reduction in anger responses. Loss of memory. Distractibility, so people are easily distracted. Seizures, which, interestingly, the clip did not, or said that that wouldn't explain the seizures. It actually does. And dementia. Okay, especially if the person is far along in age, older. I don't know why I said far along in age, like it's some sort of disease. Aging is a disease. So these are the pathways that so these apply to the amygdala that I just showed you. But specifically when it comes to fear and how, specifically, what do I want to say? Specifically when it comes to like the Cosmides and Tubi idea of these systems and subsystems that have been programmed over and over and over again from generation to generation to generation. As you can see, I take a very hard evolutionary approach to my description, discussion, and exploration of emotion. I find it to be the most compelling of explanations. Of course, other psychologists would would tell you differently depending on their backgrounds. But because my background is more along the lines of this stuff, it's the most compelling, right? It's the one that seems to be the most explanatory for why our structures do the things they do. Okay, and so fear conditioning is one of those things. And so I wanted to share the pathways. So in chat, let me know who's not familiar with classical conditioning. Let me know if anybody, so just let me know in chat if you're not familiar with classical conditioning. Because if everyone is familiar with it, then I can kind of skip going over it. I take it that by the lack of response, everybody is familiar with it. Familiar with the properties of classical conditioning. Because, I mean, the slide itself has acronyms on it. So it's like, if you're not familiar with what those acronyms are, then they're not going to be... they're not going to be helpful. Yeah, Pavlov dogs, Julia. Yeah, definitely. Yeah. No, we're on a bit of a delay, so if anyone is still thinking about it. Alright, so I think the silence there, I can go ahead and skip through that. So here we are in the brain. Okay, so this is the anterior temporal lobe right here. Okay, so you can see how close the amygdala is to the end of the temporal lobe. This is the very most anterior part of it. So this is the front part of it right here. Okay, so you can see that it's buried sort of in the middle, and then you can see those structures that I mentioned. So you've got the basolateral, the lateral, and the central. Okay, basolateral B. LA is lateral, and CE is central. Okay, and so you can see here that behavior, ANS, hormones all come from CE. So central sends out the information. So just to reiterate, central sends out the information to the various places that it needs to go, and the basolateral and lateral areas receive information. Okay, so receiving information, sending information. Okay, so that's how. So these receive input from various places, like the hippocampus, which is this blue structure right here, this fading out blue structure here, and then the central sends out stuff either back up to the cortex or down into the hindbrain, midbrain, other areas, etc. Okay, so let's explore how fear then is, and specifically conditioning, right? So connecting a once neutral stimulus to a response, a reflex, an elicited response. Elicited. Yeah, that's the right word. Elicited response that we have that is reflexive and we don't think about. Okay, so salivating when food is present, as a la Pavlov's dogs, right? So fear conditioning works in that way, right? So we have the neutral stimulus, which becomes then the conditioned stimulus, the thing that is fearful. Okay, and then the conditioned response is now the fight or flight response, specifically, and then the things like trembling and being afraid, fearing for my or worrying about my safety, having anxiety, that sort of thing. All of those coalescing things around fear, right? So the unconditioned stimulus, the US here, goes into the thalamus. From ears, eyes, nose, well, maybe not nose, but, mouth, whatever, but mostly ears and eyes. We'll just say that. So it's coming in from the thalamus. It's a switching station, right? Which sends feedback to the somatosensory cortex, which is, up top here on both sides. And that all goes into the lateral amygdala, okay? And, jeez, it's orange. Okay. Oh, still orange. Awesome. Stupid camera. And then behavior then goes from the lateral into the rest of the brain for various things, right? So let's say a bear chomps at you or starts running toward you and you're like, wow, that bear's getting bigger. I don't like that. That bear's getting real big. I think it can hurt me. And so the more times you see a bear, the more you are afraid of it. Or you got mauled by a bear and you're like, oh, well, maybe I shouldn't touch bears anymore. I don't actually want to use the bear example. Let's talk about bees. Who's afraid of bees? Or, more importantly, who doesn't want to get stung by a bee? I know I don't. Actually, let's talk about wasps, because we'll leave bees alone. We'll not throw them under the bus. So let's talk about wasps, because wasps can keep stabbing you over and over and over again because they're awful, unhelpful creatures who just are the worst. All right? So let's use fear as a wasp. So here's the unconditioned stimulus, a wasp. You've never encountered a wasp before in your life. And you're like, do-do-do. Oh, that's a really interesting looking death bee. Oh, I'm gonna go up to it because I really want to examine it. Oh God, it stabbed me! Oh God, ow! Ow, it keeps stabbing me! Why does it keep stabbing me? Okay, so, that is this over here. So that information went into your amygdala. The stabbing and the pain and the look of it, and you're like, I don't ever want to see that again. And so then that becomes the CS. That becomes the conditioned stimulus, right? Over and over and over again. But sometimes with fear conditioning, it just takes the one. And so with wasps, we're just going to use the one. From that one instance of, oh, what is this? To, oh my God, it's a wasp. Okay. And so we are going to then see that this travels through the auditory stimulus. So if you hear a bzzz going around, you're like, what was that? Okay. That goes into a little thing called the ITC. These are, this is short for intercalated cells. There we go. Intercalated cells. And they connect the auditory cortex with the basolateral and lateral parts of the amygdala, which then send out, which directly, as you can see, the ITC sends information directly to the central complex, which then broadcasts to the rest of the brain. Okay, and that broadcast is, there is a wasp somewhere around here, or there's some flying thing that is gonna, and I need to leave right now. Okay. So that is what ends up happening with a conditioned stimulus. Okay. I bless Katerina. Hopefully it never happens to you. What? And you're on alert for them because you know it's coming, right? All right. So, how do we know this is a thing? Well, Elizabeth Phelps. Why is this going so slow? So Elizabeth Phelps was like, oh, how do I study this in humans that are alive? can't necessarily look at what their amygdalas are doing. So she developed a instructed fear paradigm. Okay. And so I'll show you sort of an example of this, but I'm going to explain it by I'm going to explain it verbally first. Okay. So what she did was she showed people either angry faces, okay, or neutral faces. And she just put a the skin conductance, the SCR thing, the galvanic skin response thing that goes on your palm, because your palms get sweaty quite a bit when you are in sympathetic nervous system activation. Okay. So that was the measurement. I'm not entirely sure what you mean, Julia. The skin conductance response was the measurement to responses to the angry and neutral faces. Okay? Right. And one of the reason why she used angry faces is because we don't like angry faces. These can actually become fear stimuli, right? These could be fear eliciting stimuli. If it's an angry face, and let's say it's like a big dude who's like, "Ah, I don't come here, I'm gonna eat you," and you're like, "Oh no," okay. So participants are told that the purpose of this study is to measure physiological response to pictures of human faces, and electric shocks are going to be presented. Okay, so if you were in learning last semester, you'll know all about electric shocks. All participants believe the instructions. Okay, so there was a manipulation check right after the instructions were presented. Like, do you think this is what the study's about? And they're like, yeah, sounds reasonable. The instructed learning group is told which stimuli predicted a shock. So there was one group who were told that, angry faces were gonna predict a shock. We're gonna present a shock. Okay. However, no shock was ever presented. Okay. There's it's gonna be on angry faces, but random. Okay. And so because of the random addendum to that, it never came. But results indicated that even though they were told that, or even though that they never actually received a single shock, the verbal instructions that they received through the the auditory cortex, that was processed. It produced the same emotional response of sweaty palms and you preparing to be shocked than actually getting so the same response. Okay, explicit awareness of the fear eliciting stimuli. So that's the instructed fear paradigm. And Katerina, that explains why you have a healthy fear associated with wasps, though you've never been stung. People have told you about it. These are verbal instructions. So this is instructed fear. Okay? And so you have the same responses that I would have in the presence of wasps, because I have been stung before, and one little effer when I was a kid stung me in the exact same spot twice. So, So let's, Elizabeth Phelps was on a, oh my gosh, come on. Hello, thank you. Elizabeth Phelps was on a, like a podcast sort of thing. So forgive the, campiness of some of this stuff, but, she explains more about her research, better than I could do. It's, I don't remember. Yeah, it's 8 minutes. I'm gonna fast forward through some of it to get to her. because this guy's song is,
Welcome to My Mind's Eye, where we talk about mind and brain and listen to music about...
I'm sure that would give me all the details of...
Okay.
So if I were to ask you, Joe, what were you doing 9/11? You'd have a story, I'm sure, that would give me all the details of how you first heard about it, where you were, where you went next, who you talked to, what you saw. I mean, I have that exact same experience. I know every little detail of that, whole day up until about noon. I could tell you in, minute seconds, right? The problem is that's about 50% right. Your memory for 9/11 is about 50% right. My memory is about 50% right. Now, I am sure my memory is right? But the data suggests I'm wrong about that. And then you have to ask the question, why does our confidence for highly emotional events exceed our accuracy? And exactly how does emotion change your memory?
Things that you used to say, there was a broken record in my head all day. Liz, you've been working on ways to alter memory, especially in an effort to help people control unwanted feelings of fear and anxiety. Give us a little background on this and tell us what you've been up to.
So we've known for a long time that every time you store a memory, there's a period of time where the synaptic changes that become the memory in your brain occur. And that period is called consolidation, and it takes a decent amount of time for that to happen. If we were to do something before you fully stored the memory, the best example of this is if you get a concussion. If you get a concussion, often people don't remember what happened right beforehand, and it's thought to be because the concussion has sort of disrupted that storage process. We call that process consolidation. And there was this renewed interest in this other idea that basically now every time you retrieve a memory, it once again has to go through a storage process. This sort of reconsolidation window, this was sort of reinvigorated by a study by Kareem Nader in your lab, where he was able to take a simple threat memory, pair a shock with a sound, and later on, after that memory was stored or consolidated, just play the sound, inject into the lateral amygdala, where we know these sort of synaptic connections occur, a drug that blocks that storage process. And when Kareem did this, it looked like the memory was gone. The rat no longer showed any type of fear response or threat response to the tone itself. So this was really exciting, super exciting work. We wanted to do it in humans, but we can't. We can't inject these drugs into the amygdala in humans. But this type of work suggested to us that memories are vulnerable even long after they're stored. And you can make them vulnerable again by retrieving them. And so 2 students, Marie Monfils, working with you, studying rodents, and Daniela Schiller, working with me, studying humans, got together and started to think about different ways to do this. And one of the things that they came up with was maybe memories are vulnerable in different ways. So you don't have to target it with a drug, but when it's vulnerable, if you introduce new information during that time window, this sort of reconsolidation window, you might be able to permanently alter the memory. And that's what the data suggested.
Night when we were running blind and tearing free. So let's talk about the broader implications of this work. How does it fit into the context of our general understanding of memory, and can it possibly be used therapeutically?
We kind of think of memory.
I'll let her speak a little bit more.
Layperson thinks of memory as like a tape recorder. I mean, psychologists have known this is not true. Some of the earliest, work in memory suggested that memory is reconstructed. That you sort of bring in new information with old information, and really it's like a retelling of a story. Someone once gave me the analogy of the game of telephone, right? So, you tell something to somebody else, who tells something to somebody else, and next thing you know, it's like a whole different thing at the end. This is a process, a natural process that occurs. and it occurs whether we understand it or not, right? And so the fact that we can understand it, however, and now start to target it specifically, suggests, we can think about using this in therapy. One of the ways this might work is to think about these time windows when memories might be most malleable, and then do your intervention. So I think that one of the best examples is actually a study that took advantage of this vulnerable period. They essentially replicated what we did, and they used our exact parameters and did it with addicts and showed some success. And I think that's probably the best example of translating this basic work that you and I developed to a clinical domain.
Well, I just want to forget how to remember you. Any advice?
All right, that's enough of that. So you can see there the mixture of memory and emotion, because they're specifically talking about these emotionally charged events. I realized that wasn't as clear in the video as it was to me when I first watched it. But they're talking about emotional events, the idea of 9/11 or some other flashbulb memory. And so that rat experiment with injecting the amygdala into, injecting a drug into the amygdala to prevent connections, and how that then does not reinforce memories associated with, or at least the emotional aspects of those memories. So those memories become, essentially the idea is those memories become everyday memories as opposed to these vivid flashbulb memories that many of us have for lots of things. You're probably going to end up with some flashbulb memories surrounding this this whole coronavirus pandemic. It's not a single event, which is generally speaking the case when we talk about flashbulb memories, but you'll probably have some sort of thing like when, I don't know, you heard about the first person dying, or if you have a loved one, unfortunately, that dies due to the virus, right? Or you have a friend who has somebody they know, and so there's these connections, and for the grief that you might have for lost experiences, etc. These will all go through the amygdala first, and they will then go through the hippocampus, and back and forth, while you consolidate them. Okay. Now this stuff here is the data from the instructed fear paradigm that I told you about in the previous slide before we watched the video. And you can see here that, so this is activity in the amygdala. Okay, bigger numbers means greater activity. And on the y-axis is skin conductance response. So higher numbers here mean more sweat, which means more conductance, right? Because sweat has electrolytes in it, and so it actually acts as a conductor on your skin, which is by itself really interesting. But sweat does have those electrolytes in them because that is coming from glands within your body. So it's salt. And so you can see here that as activity in the amygdala increases, so too does skin conductant response. And this is the line of best fit. It doesn't necessarily look like it on this scatter plot, but I can tell you that this is a fairly good regression line here. Okay, and here is where the activity in the amygdala is activated. Now B, this graph here, shows activity in the left amygdala when a CS is presented. So when an angry face that is supposedly going to have a shock associated with it is presented, you're just like, I'm gonna brace for it. And then it never comes, right? So that amygdala activation and the sweat comes from the anticipation of the CS. And so you have this CR. You have this CR. Because the amygdala is ready for it. The amygdala is ready for it. Okay? So, if we were to break it down from an emotion standpoint, so a lot of the things we learn are learned explicitly. Okay? Through various methods. A lot of it has to do with operant conditioning, but we understand that, and especially as we get older, that we don't like the things that don't make us feel good, and we like the things that make us feel good. Fearful things do not make us feel good. And so, while we learn these things explicitly, the amygdala is critical for that underlying indirect expression of the fear response. So we may be afraid, but we need the amygdala to actually get our body to act afraid, if that makes sense. We can say, hey, I'm afraid. I can say, man, I'm really scared right now. I'm really scared right now. But my body may not actually do anything. Because just saying the words, I'm scared right now, or this coronavirus thing makes me scared, isn't actually going to activate my sympathetic nervous system just because I can say those things. So you need both pieces. You need the cognitive bit, and you need the amygdala bit for the expression of fear. Okay? In contrast to that, I wanted to share with you SM. SM is a famous patient in the fear literature, fear and emotion literature, because she had difficulty understanding and expressing fear. Okay? So the first thing that I want to show you is when she was asked by some clinicians to draw faces that reflected the emotions that were written. So they were blank spaces, and just underneath was the word happy, sad, angry. Oops, I keep doing that. Surprised, disgusted, and then afraid. Okay, so she drew a woman with a big old smile. Happy. Great. Perfect. She drew a little kid, which appears to have spilt their milk. No, there's no crying over spilt milk. But this kid looks sad, right? It's got tears coming down its face. It's got a frown, right? The ends of the lip are pointed downward. Okay, angry. This man looks very angry. Okay, it's probably because his hat is too small for his head, but things shrink. But one of the things that she's able to do is furrow the brow, get them in here nice and tight. And then, very small mouth, that sort of thing. Surprised, open mouth. Surprise usually is... You can see that the eyebrows are raised, right? Eyes are wide, mouth is open. That's a very good surprised drawing. Disgusted? Disgusted doesn't really have a ton of facial features, although one of them is a crinkled nose, because, it's just like, which I'm assuming is what she drew here. A lot of times people will draw tongues out, like, Yeah, you know when you're disgusted. But here's afraid. It's a baby. What? That's odd. Okay. Researchers, the clinicians asked her why she drew a baby for afraid. She didn't know why she did that. She didn't know why she did that, but she knew that she was not happy with it. She knew that it did not reflect afraid. She could not tell them why it stood for being afraid. And that's because SM lacked fear response, an appropriate fear response. She's very famous for not having the ability to detect fear and feel fear. And then so here I found a video of this of a clinician talking about SM. So a lot of this stuff happened quite a bit ago. But here is this researcher talking about SM, and I think it, again, better than I could do. So we'll listen to what she has to say. It's about 2 minutes.
So SM is one of the most famous case studies in all of affective neuroscience. She's a woman who is currently, I think, in her 40s. And as far as we can tell, the destruction of her amygdala has left her essentially fearless to at least external stimuli. And to test whether SM is in fact fearless, the researchers who worked with her took her to a couple of the most frightening places that they could think of in the local area. So one was an exotic pet store, and they offered her exotic snakes to hold in the store, even though she says she's afraid of snakes. Many people would hesitate to hold a snake right up to their face and touch its tongue and inspect its face really closely, but SM had no problem doing that. They also took her to a place that's, it is at least called the most haunted house in the United States, and they decorate it up for Halloween, and they have people, the staff, dressing up as monsters and things to try to scare people. And the researchers in SM went through with just a regular group, and all the other people who were in the group, as soon as they were in this haunted house, it was apparently very spooky, were huddling together, and they were really slow to go around corners, and SM was taking the lead and saying, "Come on guys, follow me," and she would be the first around the corners. And when the monsters jumped out and tried to scare her, apparently she would scare them sometimes, because she had no fear response to them, which they were not expecting. And so what was really interesting about this is that SM is not emotionless. She experienced a lot of curiosity and excitement, which we think are governed by other regions of the brain predominantly. But she just doesn't seem to have any normal anticipatory fear response. But her lack of a fear response gets her in trouble. So she apparently walks home in the evenings through sort of a vacant lot, and she was mugged there at one point, and I think at knife point. And most people, when something scary like that happens to you and you are at risk of getting hurt, you have a normal fear response when you're approaching that same place that would send you avoiding it. I mean, that's what the fear response is all about, avoid things that might hurt you. And she doesn't have that response, so she continues to take that same route home every night by herself.
Now, not having a fear response is not necessarily going to be the end-all, be-all of anything. And it's not as dangerous as not being able to feel pain, for example. So feeling pain and feeling fear are quite a bit different, even though they have similar evolutionary underpinnings, right, to keep us alive. But not feeling fear maybe won't be as dangerous. Jessie, isn't the only thing that put fear into her was a lack of oxygen due to nitrogen or some gas being given to her through it was carbon dioxide. It's carbon dioxide. And there was a recent study in 2013 where they had a pair of twins who had damage to their amygdalae and did not show a natural fear response unless just given carbon dioxide. Yeah. It's rad though, right? It's rad because lack of oxygen. And I wonder if the internal feelings of gasping for air or not getting air is activating some other survival mechanisms that isn't necessarily fear, but it's being interpreted as fear. So it's just like suffocating, right? Suffocation's a slow, agonizing death. It's like similar with drowning, right? So it's not being able to get oxygen is essential to our, directly essential to our survival. And so yeah, that was able to produce that fear and carbon dioxide. Yeah. If you find a video, let me know, because that would be amazing. I'd love to have that. for the future. All right. A couple more bits and bobs. Well, maybe not a couple more. Where am I? Oh, yes. So I just want to quickly talk about Zorowski, and Sapolsky. So Zorowski said that acute stress facilitates memory. Okay, thanks, Jesse. Facilitates memory, so that is stressful things essentially become may become CSs, conditioned stimuli for that particular response, the stress response, anxiety, and other autonomic responses. And then, we have Robert Sapolsky's work. And so if you're in health psychology this semester, you've already you know about the Stress Portrait of a Killer documentary that you watched earlier this semester. And so you know all about Robert Sapolsky's work and how stress changes hormone levels and how those hormone levels then turn into negative health impacts like high blood pressure, advanced cardiovascular disease, and it all stems from his book that he wrote in the 1990s called Why Zebras Don't Get Ulcers. And the idea is that, yes, stress is rough, okay? But they don't get ulcers because they respond to their environment in the way that they're supposed to. They see a predator, they get the hell out of there. Otherwise, everything's gravy, baby. Everything's gravy. And so they don't get ulcers. Humans, however, have taken stressors to the next level and end up with feelings of, or these stress feelings open the door for mucus layer in your stomach to break apart and allow bacteria to then inflame and eat the inside lining of your stomach, right? Ulcers. And the idea is that this stress, the memory of the stress, the stress, is feeding into your memories. Okay, it's no coincidence. It's 100% no coincidence that the amygdala and the hippocampus are right next to each other, and why emotion and memory are so strongly linked. Just go look at Inside Out for the answer to that, right? Everything about the idea in Inside Out is that those memory orbs from Riley's life have a color associated with them, and those colors are the emotions that Riley has. And so the dominant emotion in that situation for that memory is that color, okay? So, whatever the colors were, right? And so the idea here is that our stress does also play a role in that. And we're talking about things like anxiety and among others, okay? What time is it? That's 12. I don't know. All right. Switching gears again. So that was just a little bit of a stopover. Now I want to finish today with talking about the orbitofrontal cortex, which is important for how we deal with higher order emotions. So controlling our impulses, okay? Maybe not laughing at a funeral if it's seemingly inappropriate. That idea of not laughing at a funeral unless prompted to laugh. So having those higher order inhibitions, emotional inhibitions. Also things like regrets. A longing for something that didn't occur, right? A regret. And those are negative feelings, regrets. So that's the orbitofrontal cortex. And so Antonio Damasio is the gentleman, and as well as his colleagues over the years, who is most notably tied to this. And so he appeared on some program with David Brooks.
For a TV.
For a
the
TV.
World is thinking.
One of the most famous patients you had.
So David Brooks is a columnist for the New York Times.
A successful person suffered this damage, and whose life went down the tubes. Basically, made a series of terrible decisions. Another case that has always fascinated me, the scene you describe in one of your books, one of your patients who also has trouble making decisions, and you're asking him at what point should we schedule the next appointment,
Yep.
Tuesday or Wednesday, and as you describe it in the book, he spends, I think, 20 or 30 minutes saying, well, Tuesday has this advantages, Wednesday has this advantages, unable to make up his mind.
Right, exactly. And
So these are higher order cognitive
because
processes.
It shows you on the one hand that the cognitive abilities, the ability to manipulate data and to make analysis of costs and benefits in a very cold way is maintained. And people can still do that. Well, but the thing I most liked in these patients when we asked them about restaurants, and this was really quite remarkable. And they said, "What restaurant do you want to go to tonight?" He said, "Well, we could go to this one." And they said, "But I take it that this restaurant has been rather empty recently, so that's probably a bad sign. It's a sign that the food may not be so good. On the other hand, it's true that if it is more empty, we're likely to get a table, and therefore we should go there. But then, and the thing will go on endlessly until you really feel like pounding on the table and said, well, get real and choose." But the reason why they can't choose is that they haven't got this sort of lift that comes from emotion. It is emotion that allows you to mark things as good, bad, or indifferent, literally in the flesh. And it is
Those are his
that
somatic
kind of
markers,
emotional
as you can see
impetus
up
that
there.
they are lacking. They cannot conjure up for a given situation an emotional state that would decide them in one direction or another. And of course, this is something that we all can relate to. We're constantly being swayed in what we do by just a little teeny change, something that comes, for example, from our past experience with a certain kind of situation. But what we remember from a previous situation that is like that is not just the facts, not just the outcome that it may be good or bad. We also remember whether or not what we felt was good or bad. And this is something that people need to understand, is that when you are making decisions any day of your life, and of course the options you make are going to produce a good or a bad outcome or something in between, you do not only remember what the factual result is, but also what the emotional result is. And that tandem of fact and associated emotion is critical. And of course, most of what we construct as wisdom over time is actually a result of cultivating that knowledge about how our emotions behaved and what we learned from them.
So that's the idea behind somatic markers, these biological underpinnings for whatever emotions that we have that then indicate higher order cognition, like decision making and impulse control, and complex emotions like sorrow, regret, elation, spiciness, complex emotions, right? One of the things that Damasio and colleagues' work is most famous for is the Iowa should say gambling, not gabbling, gambling task. And if you were in learning last semester, Katie tried to win some money. Go to this link if you want to try to beat the system or determine what the pattern is. So the Iowa gambling task is essentially a gambling task, obviously, but there are rules associated with it, and the rules are incredibly easy to determine if you have a functioning orbital frontal cortex. And orbital frontal cortex is essentially the cortex that is just above the eyes, okay? And so it's just in front, but on the ventral end. Excuse me. Didn't you lose money, Katie? I think you lost money. Because you start with some money, and then I think you ended with less than that. I don't remember fully, but, so once you have that pattern and what you should choose, then you can start gaining as much money as possible, right? You can just like, boom, 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 I'm doing super great. People with OFC damage, however, cannot determine the pattern. They cannot determine the pattern because they are struggling to determine what's good about winning and what's bad about losing, okay? And so they have the inability to deal with that. And so the gambling task has been used to determine whether or not somebody who has symptoms associated with OFC damage actually do have OFC damage, which is great. So a question for you in chat as we end for today. What regrets do you have? What are your regrets? Leave them in chat. I'm curious. Now, obviously, don't put a personal regret that you don't want to share publicly, but if you have any regrets that you would want to share publicly, put them in chat. I'm curious. Let me know. Your regrets. No regrets. No regrets. Okay? What are your regrets? Not buying the one Funko Pop at Hobby Lobby when I could have? Yeah, now they're closed. It's a bummer. Sorry, Julia. Okay. Okay. Catwoman. Not bad, not bad. What else? What else? Come on, everybody. Get going. Let's move it. Move it or lose it. Katie, not bringing my car back when I left campus before break. It's now sitting in the Arnold parking lot. Oof. You regret your past procrastinations and current ones, Max? I'm so sorry. Well, at least for the past procrastinations, you can drop those, right? Katie, you should probably go get your car. I'm sure somebody could drive you there, right? Austin, you don't have any regrets? So you tattoo no regrets on your arm? No, maybe your neck. No regrets. Yeah. All right. I mean, that's fair. That's fair. Yep, no regrets as well. No regrets. Come on, others. Come on, everybody else. Let's go. I want to hear it. Tattooed, no regrets. Yeah. Getting stuck down a muddy road when I was just trying to drive around and enjoy a night drive. My dad was none too happy. Oh, I'm so sorry, Jesse. That sounds rough. Was that recently? That sounds rough. Especially since you incurred the wrath of your father. No regrets. A little over a month ago. All right, well, you know. I guess that was pre-COVID lockdown. Who else has some regrets in chat? Drop it down, drop it down. Ooh. It's almost time for lunch. I can smell it. I regret not getting more regrets in chat. I regret that very much. Did that work? Did that make you feel guilty? That's another emotion. I have too much anxiety to try to think of my regrets. I'm sorry, Deanne. Maybe you regret having too much anxiety. I do regret not bringing Brian home. Yep, I do regret it considerably. Yep. So much. I regret showing my wife how to play Jurassic Park Evolution. Now I don't get to play when she gets home. Oh, no. Marital strife. Oh, no. Stayed at a friend's place not to have to deal with waking him or up around midnight. It just made him angrier. Oh well, live and learn. Alright, that's fair, that's fair. Jurassic Park Evolution. That game looks pretty cool. I worry about the replayability of it though. You know? Alright, that's fair. W for the marriage, personal loss. Yeah. You gotta get a computer that can handle it. Another computer that can handle it. You know, get another computer with some decent specs. Yeah. All right. Well, yeah. We're not ending until I see more regrets. I want to see all the regrets. More regrets. More regrets. Regret not saving more money because I'm going broke with this virus. Yeah. Oh, I'm so sorry to hear that. It's rough. That's rough. I hope you get at least get the stimulus super quick. All right. So there are 3 more slides on this, but I think I'm done for today. You can review those things. They're in the book. Whether or not you use the information, for your final is a whole other thing. So if you have any questions, just let me know. Cameron, I regret getting 3 drinking tickets. It's cool. Ooh, dude. Yeah, that's a bit of a bummer. Jesse says, by the way, it wasn't completely about SM, but the video I watched was he was talking about it. I have set it to start where he is talking about SM. Okay. I will watch the whole thing, though. Let me see if I can... Nope. Alright, well, I had to click it. That's annoying. Should be able to copy paste it. Julia says, I regret not visiting my crap Rusty before the break. I'm so sorry.
What about people who don't have...
Ah, it's Vsauce guy. Julia, I regret not visiting my cat before a break. Now I know when I'll see him. He lives with your grandmother. Oh, I'm so sorry. Yeah, that's a bit of a bummer. Abby, staying on campus during this time because it is very lonely. Yeah, because you can't go hang out with people who are also on campus. You basically are confined to your dorm room. Oh, I'm so sorry. It's a bummer. That is a bummer. Hopefully there's outside time that you all can do from 6 feet apart. We'll eventually get there, right? Okay, well, I think that does it for today.